Hey, in this tutor we will discuss about movement across the membranes, different type of movement, how they occur, and what are the particular type of protein that are helping in these particular type of movement. First of all, let's discuss about simple diffusions. Now let's see that if our temperature is not below absolute zero, like 273 degrees centigrade, all the substances or particular type of gradient of particular type of substance will move randomly from the area of high concentrations to an area of low concentration. This is called simple diffusion, like movement of substances from higher concentration to lower concentration by random movement. Their random movement. Substances move from higher concentration to lower concentrations randomly until both the concentrations on both sides become equal. We will discuss about this one. It is very slow process and it occur at short distances, and cells that are most closely combined with each other, and it also occurs from high concentration to low concentration, and it do not require energy. Like it is not an active process, it is a passive process. Active process are those processes which require energy to transfer materials from one side to another side, and they are against the concentration gradients from the low concentration into high concentrations. Then there are facilitated diffusions, like simple diffusion in which molecules move from one side to another side, here the concentration of substance is very much high, and here the concentration of substance is very much low. The substance will go from higher concentration to low concentrations without any hindrance or barrier. It is a simple diffusion, but there are many examples where simple diffusions cannot happen, plasma membrane. Plasma membrane is a lipophilic molecules, and it will not allow an hydrophilic molecules to pass through it. So in particular type of situation we want a carrier that will move substances from higher concentration to lower concentration, and this type of movement in which carrier substances move a particular type of substance from higher concentration to lower concentration is called facilitated diffusion. Like we will understand the concept of transport protein. Like outside is the extracellular fluid, there is high concentration of polar molecules, and inside there is a low concentration of polar molecules, but polar molecule cannot simply diffuse by this plasma membrane because it is hydrophobic, and it will not allow a polar molecule to pass through it or any charged molecule to pass through it. So a simple diffusion will not work in this particular type of scenario. And now we will have another type of transport in which we have a transport protein or protein channels or pores that will allow a substance from higher concentration to low concentrations. These protein channels, in which protein these are the protein channels, and the movement name is facilitated diffusion, that are facilitating a particular type of movement. There are various type of movement like carrier protein or pore. These are the pores that will help transferring substances from higher to lower concentration. There are the carrier proteins, they act as differently, any particular type of substance combined to them, and then intracellular domain, here it is closed, then it open and substances move into the cells, and they remain closed by the next times. Then we will discuss about osmosis. Osmosis about the movement of the water molecules and movement of water from its higher concentration to lower concentration is called osmosis, and we will discuss it with respect to solute concentration and different type of other carrier proteins, like water molecule can easily pass through the plasma membrane, though it is a polar molecules, but it is a small and uncharged charge is very little much, so it can cross easily the plasma membrane. But there are also many substances in the plasma membrane, like the carrier protein like aquaporin, that will transport substances from one side to another side and transport substances from higher concentration to lower concentrations, like these there are many various type of aquaporins. They are numbered from aquaporin 1 and 2, like you will understand that thyroid, that vasopressin hormones that act on the distal convoluted tubule and absorb water, and they express various types of aquaporin protein, and aquaprotein channels then transport water molecule from higher to lower concentration. Tonicity. What is meant by tonicity? Tonicity is the concentration, simply the concentration of, if we define the tonicity, it is simply the concentration of a particular type of solute in a given solution, and like if the particular type of solute and particular type of solute concentration in environment is similar to the tonicity of cell, it is called isotonic environment, in which water move inside and outside of cell at equal ratio, and so cell shape remains same and cell size remains same. Equal amount of water will be ejected and equal rate of water will be move inside, so it is will be balanced. Then hypertonic environment, an environment in which solute concentration is higher in outside and water concentration is higher inside. In this scenario, water will move out of cells and it will cause the constriction of waters, and it will cause shrinkage of waters, and this condition is called crenation in the red blood cells, in which there is shrinkage of red blood cells. So this is the red blood cells, and it is placed in hypertonic environment, then water will move out of it and it will cause shrinking, and this shrinking is called crenation. It is very important. Then there is hypotonic environment, in which there is more of the water concentration outside and more of the solute concentration inside. This will cause the movement of water inside, and it will again reverses the size and will increase the cell size, like water is moving inside and less water is moving outside, its size will increase and will cause bursting of the cells or particular type of RBCs. So here is our discussion is ended. We have discussed about simple diffusion, like molecule move simply from higher concentration to lower concentration, like this is the cube of you may call it sugar or call it salt, and this is the water, in which concentration of sugar is very much less. Then there is that movement of water molecule randomly from higher concentration to lower concentration until both the concentration become equal, and this will continue after this and will stop at equal concentration. Then there are facilitated diffusion, in which carrier molecule or ion channel will move molecule from higher concentration to lower concentration. Then we have discussed about osmosis. It is about the water molecules, movement of water molecule from higher concentration to lower concentrations, or from hypotonic environment to the hypertonic environments, and like this one is isotonic environment, in which the normal cell size is maintained. Then there is a hypertonic environment, shrinking will occur and crenation, and in a hypotonic environment there may be bursting of cells. So we will discuss about filtration later on.